Hey everybody. Alright, so we're moving on to one of our last topics. Antibody drug conjugates is an anti-cancer therapeutic strategy. It's pretty cool because it has some organic chemistry and some biology, and it's a pretty easy concept to grasp. And we'll go through one really good promising example of this produced by the company Genentech on an antibody drug conjugate that they're probably going to put out into the clinic after clinical trials. So what are the things we're going to talk about are just a couple quick examples of antibody drug conjugates in very broad terms to get us kind of on the right page. And then we're going to talk a little bit about anti-mitotic drugs, because a lot of the antibody drug conjugates, at least that target cancer, work through an anti-mitotic pathway. And so we need to sort of review how, I'm not going to go through all the details of mitosis, but kind of how some of these drugs work, like Taxol and vinblastine and things like that, in a to prevent mitosis, and that's a common target for anti-cancer drugs. Well, then I'm going to talk about the synthesis of tubulysin, which is one of the warheads. It's like a small molecule thing that can be attached to an antibody that actually has an anti-mitotic action. And so this will be a little bit of chemistry, but it's not that hard, and we'll get through it. It's a Organic Letters 2009 paper. And you're totally welcome to read these papers, and I would recommend it. I'm going to put them on the website for you to check out, the Organic Letters, and also the key paper in antibody drug conjugate that was produced by Thomas Pillow's lab at Genentech. And this paper will be called Stabilizing a Tubulysin Antibody Drug Conjugate to Enable Activity Against Multi-Drug Resistant Tumors. And this was a couple years ago in the ACS Medicinal Chemistry Letters. It's a really nice paper, and it's pretty understandable, but probably good that I'm going to help you guys get through it. And all right, let's get started. Okay, so a couple quick examples of ADCs, just to you can kind of understand what the normal structure of them is and how they work. Brentuximab vedotin, it's also called Adcetris. It's a commercially used anti-cancer drug. That's a ADC. So these will have a couple components to them, and one thing is the antibody part, and then this can be linked to a small molecule somehow. Okay, and so this shows sort of how it might look. The MAB is the antibody, and then there's going to be some kind of attachment point, so it gets attached, to the small molecule. This is a protein, and we want to separate our substance a little bit from it. So there's kind of an attachment point, a couple carbons, and then like some kind of linker that can be cleaved. And often people have been using valine next to citrulline, which is an amino acid. And this is cleavable by a protease called cathepsin, and this is present in the lysosome. And so it'll get into the cell, and then this will be kind of cleaved off, and then the spacer unit will be also it can also just sort of destroy itself, and then this will leave you with what we call the warhead. And the warhead is, in this case, it's MMAE, which is one of the several warheads being used. This is popular, but it's not necessarily the best one. And the paper we're going to talk about in a few minutes will be a kind of an improvement over this. Okay, but this does inhibit mitosis, and it is cytotoxic for cancer cells. There are some issues we'll get to later. Okay, so the linker component is this is the valine citrulline piece. It's cleaved by cathepsin, which is just a protease, and it cleaves this sort of pseudopeptide linkage we have right here. And then we have a spacer unit, which is this 4-amino benzoyloxycarbamate, which is this will kind of get expelled after the cathepsin unit is cleaved off. And then there's the warhead, which is monomethyl auristatin, or MMAE, and this is a cytotoxic peptide that binds to tubulin, and it kind of inhibits tubulin polymerization. And in a cancer cell, that's a problem because, well, cancer sort of depends on out-of-control cell growth, and it uses tubulin to do that because that's how cells grow. Okay, so this is a clinically used ADC, and it was approved in 2011 to treat Hodgkin lymphoma, which is a white blood cell related lymphoma, so the cancer of lymph nodes, and also anaplastic large cell lymphoma. So these are all different kind of variations of lymph node cancer. The second class have to do with T lymphocytes, which are another class of lymph node cells. And also in 2017, this was approved to treat cutaneous T cell lymphoma, which is lymphoma under the skin. And this is actually a well-selling drug. It's fairly recently approved, and they're earning something like $600 million a year. And eventually it's expected to peak at about $2.7 billion a year. So you can see that this is a profitable area of drug development. All right. Okay, a couple more examples of antibody drug conjugates that are released on the market. This is gemtuzumab. They all have fun names to pronounce. Ozagamicin. Probably mispronouncing that. So it has an antibody part to it. There's a linker. This uses a hydrazone linker, which is cleaved by aqueous hydrolysis, I believe. And then this uses something called caliciamycin, which we have not gotten to talk about in this class, but caliciamycin is a enediene component, and this sort of cyclizes to make a benzene diradical. So it's a pretty easy electron pushing mechanism, and when you have a enediene, it does that, and inside the cell that does a lot of damage, and because you have radicals, it basically creates aromatic radicals which just rip off atoms left and right and destroy cells. Well, that would be really unfortunate if this was destroying normal cells, but with the right antibody, this targets cancer cells. So this actually looks for markers on the surface of the cell that it can connect to and internalize into. So this is really a drug targeting device, the antibody, or like a tumor targeting device. Okay. Also, it says random lysines, but I think they meant to say random lysines. This paper it's actually a book, Predictive Markers in Oncology, 2018. I think it's kind of an expensive book, but it is one of the sources on antibody drug conjugates. There's a lot of published literature in the peer-reviewed literature that you can just download. Okay, so this is also called Mylotarg. Another example is trastuzumab emtansine. So this one has a warhead. It's kind of connected through this linker unit to the antibody. This, I believe, is another anti-mitotic warhead. Another thing to look at, let's go back to this one, the little hexagons. This kind of shows where the drugs are actually localized. And also, back to this, it says random lysines. What that means is there's various lysines that are nucleophilic on the antibody, and this attaches to them kind of semi-randomly. So it's in various places around the antibody, and that's kind of not very homogeneous. So it can, you have the drug and the linker sort of scattered at various places. This does the same thing. It's attached at random lysines. So you see the nitrogen linker, and that's kind of how you know it's a lysine-linked ADC. Okay. And this is also, I think it's Kadcyla. It's another antibody-drug conjugate that's commercially available. These are a couple more. Brentuximab vedotin, which is the one I just showed. This is connected to MMAE, which is an anti-mitotic peptide. And these are connected to cysteines. So the linkage is through a sulfur, and there's a linker, and then this is the part that's cleavable by cathepsin, the protease protein enzyme. So it cleaves off here, and then this thing kind of falls apart, and then you're left with the warhead, and that actually does the damage and stops mitosis. This is considered a second generation one. It's a protease cleavable linker, and it's attached to histines on the antibody. And there's 4 of these cytotoxic molecules per antibody. And then there's another one. This is a third generation one. This is actually more closely related to what we'll be talking about later. It uses tubulysin, which is a kind of a modern third generation antimitotic peptide that also binds to tubulin and prevents mitosis. And this is also this one is linked through a malato capropyl linker, and this is attached through a several carbon chain here, and it's also attached to cysteines. Okay, and so the one we're going to be showing later kind of looks like this. It's going to be a little bit different, and it's a one that's also kind of in development by Genentech. And yeah, this is Adcetris, which is this one I don't think is on the market right now. Medi4276. Okay, so how do these kind of work? This shows how an antibody-drug conjugate is sort of floating outside of the cell, and it finds it's usually targeting some marker on the cell. And fortunately, cancer cells often have a protein that can be targeted by an antibody, so it actually attaches and it kind of specifically targets a protein. I think one of the ones is CD22, which is a protein on the surface of the cell, and then the antibody-drug conjugate gets each internalized. It actually gets, along with the cell surface protein, gets internalized into the cell. The antibody will be destroyed, and kind of through the process of this, the cleavage reaction will take place, and that'll generate the drug, which you can either bind to DNA or attach to microtubules and cause some sort of cytotoxic effect. And then there's also and then the cell surface protein can be recycled in this process and then externalized again. There's other drugs that are non-antibody drug conjugates. So it's like a small molecule ligand, it's attached to a linker, and then some sort of payload or warhead that can also attach to proteins and then be internalized, and then the payload be released. We're pretty much just going to focus on antibody drug conjugates today. Okay, so how do antimitotic drugs work? So this kind of goes back to basic biology, and I'm not going to talk too much about mitosis, but just show a couple figures, how it works and how it involves microtubules and microtubule polymerization, because that's what we're basically inhibiting with these peptides or drugs like vinblastine or Taxol. Okay, so how do antimitotic drugs work? Let's talk about the cell cycle and mitosis very briefly. So the cell cycle has a couple of stages. It kind of begins with generation of a new cell after cytokinesis, and the cell enters a phase called interphase, which the cell basically just grows, and then that's followed by a phase called interphase, which is when DNA starts being synthesized in large quantities. And then there's another cell cycle phase called G2, which is another cell growth phase. So it goes from G1 to S to G2. And at that point, mitosis is initiated, and this will split the cell into two different cells, two daughter cells, and then basically go around in a circle. Okay. This is a miniature version of kind of how mitosis actually works. So during interphase, all of the DNA is replicated in the nucleus. This enters prophase, where the different chromosomes start kind of getting separated, and then there's these centrosomes in metaphase, the next stage. And the centrosomes start forming a mitotic spindle that's separated, and the chromosomes get attached through these things called kinetochores that are attached to the mitotic spindle, which is uses microtubules to accomplish this, and the chromosomes just kind of get attached, and then later through kind of a mechanical action, the chromosome pieces get separated, and then new cells get generated in telophase. So it goes from interphase to prophase to metaphase to anaphase, and the chromatids get kind of separated, and then new cells get generated. So it's a pretty amazing process. I'm always fascinated to see this. But anyway, this relies heavily on microtubules polymerizing and kind of creating a mechanical force that separates these two copies of the chromosome. So cancer is a huge topic, but in a sentence, it's a condition of abnormal cell growth caused by various factors, environmental and genetic. And there's different kinds of tumors. There's benign tumors and malignant tumors. Without getting well, we don't want to spend too much time on cancer, but I just want to mention that microtubule targeting is one of the ways that a lot of anti-cancer drugs work. And so one anti-cancer approach would be to disrupt the construction of the mitotic spindle and really the polymerization of tubulin. The drugs do this and bind to and inhibit assembly of microtubules. Some drugs actually activate assembly of microtubules, but in a way that is non-productive. It's sort of they have to be activated in a certain process for this to all work. So I think Taxol, for example, does not inhibit assembly of microtubules. It activates it, but in an uncontrolled way, and that kills cells as well. Okay, and so this just shows an example of what it looks like. This is the mitotic spindle and the chromosomes. I think, what phase is this? This is one of the early ones. This is around metaphase, I guess. And so the green lines are microtubules, and then the blue components are the chromosomes, and the little pink dots are kinetochores, which are the attachment points for the that the microtubules rely on. Chromosomes are blue, and the kinetochores are the pink little dots, and these are attachment points for the growing polymers. Okay, so some examples of antimitotic drugs. Some of these are clinical anti-cancer drugs. They all pretty much have really cool structures. As an organic chemist, I've always been fascinated by really cool organic structures with a lot of dense functional groups and chirality and complicated architecture. So this has been vinblastine. This is a natural product, antimitotic drug that binds to tubulin. Vincristine is another one. Very, very dense arrangement of functional groups. These are related. These are called vinca alkaloids. It's kind of a class of drugs that have a sort of a similar structure. There's some differences. This one's actually semi-synthetic. It's vinflunine, and one way to know it's sort of semi-synthetic is it has fluorines in it, because natural products usually don't contain fluorines. So this is one of the ones that was slightly modified from the natural forms of these alkaloids. Taxol, or paclitaxel, is another one. It's got a pretty dense organization as well. It's used to treat various kinds of cancer, and prostate cancer, I believe, and it was isolated from the yew tree, I guess, Pacific yew tree, synthesized, and I think all of these have been synthesized at one point or another. These are amazing accomplishments in natural product synthesis, because definitely not easy molecules to make, and all the chirality, every one of the chiral centers has to be set synthetically, which is a very, very is very cool how that works. I could read about that and never really get bored of it. Epithylones are other macrocyclic molecules that may be a little less complicated than the other ones, but these have a couple chiral centers, and these also bind to tubulin and have an antimitotic effect. The halichondrins, another kind of class of pretty crazy molecules, a lot of chiral centers and these weird spirocenters, the little carbons with multiple oxygens on them. There's a lot of drugs that have that kind of arrangement, and a lot of chirality, a lot of structural complexity. Cryptophycins, another class of molecules, and these have kind of different subunits that are color-coded in this example. That's not that important, so we're not really dealing with it. And then there's these kind of cytotoxic antimitotic peptides. So these have more of a peptide structure. Pretty simple. What's nice is they can be made very easily. These are not easy to make. I mean, we can make them, but not in really clinically useful quantities. Taxol can be semi-synthesized and produced from the natural source, but nevertheless, this can be made on a multi-gram scale very easily. Maybe 3 or 4 steps of organic synthesis, which is really cool. So this is MMAE. This is the one I mentioned earlier, monomethyl auristatin. It's a second-generation antibody-drug conjugate warhead that is still used, and it works well for a lot of things. It is cytotoxic and treats can treat cancer. Certain kinds of cancer, like multi-drug resistant cancer, this is not as potent with. And then this one is a third-generation antibody-drug conjugate warhead. This is kind of the focus of my talk today. And so it's another pseudo-peptide structure. It's derived from a natural product. And And there's a couple components to this. This seems to maybe be a little better for multi-drug resistant cancer. And yeah, anyway, so probably okay to consider this second generation and this third generation. That doesn't mean the second generation one's not useful, because it definitely is still used clinically, and it's making something like $600 million a year, so that's good. Okay, so tubulysin binds to the vinca binding site of tubulin, which is where all the vinblastine and vincristine natural products bind to. It's a pretty well characterized binding site. There are several binding sites in tubulin, and this shows a X-ray crystal structure of tubulysin, actually bound, and remember that tubulin has kind of alpha and beta forms, and they sort of stack in this kind of manner, and there's other proteins involved in tubulin structure, but it kind of sits in between alpha and beta forms, and I think it might go every other one or something like that. And this, or maybe it's on the top of beta tubulins. That's right. Yeah, on the top of beta tubulins and the interface with alpha tubulin. And so this X-ray crystal structure is 5KX5. You should be able to visualize it yourself. There's tubulysin. I forgot which one they used in this example, but there's just this kind of peptide protein interaction that it just sits there, and this kind of affects the mitotic spindle formation. And it's the same site where vinblastine binds, which is a pretty crazy different molecule, but there's some, apparently the receptor can accommodate both of these molecules. Okay, so these other molecules we talked about, Taxol and vinblastine, they all bind to different locations on tubulins, beta tubulin and alpha tubulin molecules. And this kind of shows a couple of the different binding sites, and they all sort of bind differently, and there's additionally molecules that inhibit polymerization and promote polymerization. Now, they can all be cytotoxic by different mechanisms, because tubulin polymerization has to be carefully orchestrated for the mitotic spindle to work. So even though these inhibit vinblastine inhibits polymerization, and Taxol, for example, and lolimolide promote polymerization, they're cytotoxic in different ways. But it's all, even though they affect polymerization differently. Okay. Another thing is that this is inverted relative to the last case, the last slide I just showed. So we were showing that vinblastine, for example, kind of binds on the top of the beta form. Well, that's because it's flipped over. Okay, just wanted to mention that. Anyway, in terms of colors, vinblastine is the yellow molecule, and colchicine is the orange molecule. These are all different sort of binding sites for small molecules. And then for molecules that promote polymerization, lolimolide is red, and Taxol is green. Where's the green one? It's right there. Yeah. Okay. So yeah, different binding sites on tubulin, and these molecules seem to have different effects, but they're all cytotoxic. Okay, so we're going to talk about this molecule called tubulysin, and we're going to step through a synthetic paper, actually, but this should be within your grasp. And it's a nice organic letters paper. So Organic Letters is one of the organic chemistry journals that's pretty well recognized, fairly prestigious, I would say, for sort of small bits of work. And yeah, you should try to read it. And then I think we're probably going to hold off on the last topic for the last lecture, because this can take a while, and I want to get through the whole thing. So let's talk about this organic letters paper. The first total synthesis of tubulysin B. What does total synthesis mean? It means that these people synthesize the molecule from scratch, essentially. Okay. All right. So this is Organic Letters 2009. These are some chemists from the Leibniz Institute of Plant Biochemistry and the Department of Organic Chemistry in Weinberg. It's a place in Germany. This shows the molecule. So this is one of these tubulysins, and this is reported as the first total synthesis of tubulysin. We'll step through the key synthetic steps, and I'll try to kind of explain it. I don't really have a chalkboard in front of me, but just for you guys to just have the basic idea of how this works. I mean, this looks like a pretty complicated molecule, and I know not all of your synthesis skills are perfect, so a lot of these reactions are really simple though, and they're based on what you already know. Okay, so retrosynthetic analysis basically means that we're going to break down the molecule, the final molecule, into its key starting materials, I guess, and then the forward synthesis, the actual synthesis that they use, will be going forward from those small molecule pieces. Okay, so this is the final product, tubulysin B, and the way they approached this, they made this bond last. So this is the last bond in the synthesis. So the little squiggly line, that shows that this is a disconnection. So as we do our retrosynthetic analysis, we're going to imagine this be chopped at that point, and then the next components will be the two pieces, which is a carboxylic acid. This carboxylic acid will happen to be attached or disconnected at this point in the next step, but we're going to focus on the right side right now. So yeah, it's kind of like this sort of pseudo-tyrosine type intermediate with a hydroxyphenyl right there. And how did they make this? Because this is not, it looks like tyrosine vaguely, but the connectivity's not right. This is not commercially available. They actually have to make this. And with the way they ended up doing it is making it from this kind of protected thing that kind of looks closer like to tyrosine. So it's got a Bach protecting group that you guys you know what that is, right? It's cleaved by acid. So TFA or HCl will cleave the Bach group and deprotect it and make the amine. Yeah, and then this essentially looks like it's tyrosine, except it's reduced, right? Tyrosine would be the carboxylic acid, and so it's tyrosine, which would be the carboxylic acid, but this is a primary alcohol, and I'm pretty sure they were able to buy this or make it like within a step or two from commercially available stuff. And then they use a benzyl ether protecting group on the oxygen here. A benzyl ether is oxygen, CH2, phenyl. So it's it's easy to remove those. It's H2 palladium takes it off. So this can be removed with H2 palladium. It's a simple protecting group for a phenol, for example, the ether here. Okay, what you'll find in their retrosynthetic analysis and the way they actually approached this was they oxidized this part to an aldehyde, because that's easy to oxidize, right? Alcohols can be oxidized to aldehydes. And then they attached this part kind of through something called a Wittig reaction. It's going to be a variation on the Wittig. It's called a Horner-Wadsworth-Evans. It's just it's like a Wittig reaction. Wittigs create alkenes, right? So if you imagine making an aldehyde and then the Wittig creates an alkene, Well, they basically did that and a little bit more, and they were able to get this. Okay. Okay, and then this piece. Let's focus on this piece now. Now they're going to disconnect this bond, so they're going to make this from like a carboxylic acid, right? Carboxylic acid type derivative, and then an amine, and then it's just a simple amide bond formation, right? Okay. This is a little bit troublesome because this bond, when they're trying to create this bond, it's a little congested with these this alkyl chain here. Looks kind of like a isoleucine type piece. It's congested, and when you have an amine, a congested amine, and you're trying to make a amide linkage, that's actually kind of tough. So they did something kind of cool. I've only seen this in the antibody drug conjugate world, but they actually use an azide, N3, and then they use this with a weird leaving group. It's pentafluorophenol, so it's 5 fluorines and that thing's pentafluorophenol, right? And it's just a leaving group. It's just like a chloride or some kind of leaving group. So this is considered a leaving group. So, but this is not a nucleophile, right? Azide's not a nucleophile. You would need an NH2 to attack this, right? Well, they create an NH2 by reducing this with H2. They use H2 palladium, and that reduces the azide to NH2. And then they have a nucleophile. Nucleophile attacks, kicks this off, and that actually creates this. Really cool strategy. Okay, and then the other stuff, a little more retrosynthesis. They snap off this piece, and they snap off this piece. And essentially, this is their fundamental starting material, which is they are able to make or purchase pretty readily. So in the end, their starting materials, we'll call them fundamental starting material number 1, which is kind of a tyrosine derivative, and fundamental starting material number 2, which is this piece that's, it has a couple of the chiral centers. It's got an acetate. Acetate, of course, is this, which is present in the final product. And it's got kind of like a valine looking thing, isopropyl group here. Okay. All right. So let's talk about starting material number 1. Let's go back to it. It's this. It's the kind of tyrosine looking alcohol. It's actually kind of like an amino alcohol. It's a Bach protected amino alcohol. So it's a Bach protected amine, and yeah, we might call this a amino alcohol. Because if you didn't have the Bach group, it would definitely be an amino alcohol, which is just a class of molecules. All right, so let's start from there. And so we're going right out of the organic letters paper right now. They did something called tempo oxidation. Tempo is it's a very weird molecule. It's probably cool enough that maybe I'll show it. What's tempo? Tempo molecule. There's tempo. It actually shows it right underneath. It's a weird red crystalline solid. It's a rare example of a stable free radical. So it's a nitrogen-oxygen radical. You can purchase it and hold on to it indefinitely. It's pretty much indefinite shelf stability. Very rarely are radicals stable molecules. Through a pretty cool reaction mechanism that you can use this to oxidize alcohols to aldehydes. And it's a catalytic method. You use very little tempo, and then there's some sort of like bleach or some oxidizing agent that actually carries out the reaction. So no, we're not thinking about mechanism. This would be something you'd learn in an advanced organic chemistry course. But yeah, it just makes an aldehyde. You could think, you could use PCC. PCC would do the same thing. PCC is kind of toxic and bad for the environment. That's why people use these non-PCC reactions like tempo. Okay, and then this is the part that's kind of like a Wittig. Technically, if you review organic 1, I think this is an organic 1 reaction. This is in resonance. This is a resonance with a carbon negative. So that is in resonance with phosphorus positive, carbon negative. The carbon negative attacks the aldehyde, like a Grignard almost. And then there's some little bit of mechanism where the phosphorus gets lost and oxygen gets lost. And then it's essentially, though, an alkene forming reaction. And they make this. So they went from an alcohol oxidized to an aldehyde, electrophilic functional group. And then they did this kind of Wittig-like reaction. Technically, the name of this is called a Horner-Wadsworth-Emmons reaction, which is Wittig-like. And then they have this. So, but if you remember, what they want to do next is convert this into an alkane, and they reduce it because it's they need to reduce the alkene, and you can reduce an alkene with H2, right? So they reduce it with H2 palladium on carbon, and that actually creates a mixture of products because you're forming a stereocenter, and the stereocenter that you're forming here is kind of mixed. You would think that maybe when H2 palladium does this, that the neighboring chiral center might have an effect and say, oh, well, this is just going to be 6A or 6B. That's not the case, and there's not very much control. That's a reasonable thing to predict, though. When you have a neighboring chiral center, you might say, oh, well, that might influence where H2 is going to go. Is H2 going to go from the top, or H2 is going to go from the bottom? Well, sometimes you get that, sometimes you don't. They did not see that, really. I think it was like a 2-to-1 mixture of these, and I don't remember the exact ratio. It's in the paper, though. You can read the paper. Oh, yeah, what else happens? It also deprotected the benzyl ether. So we had the benzyl ether, it's gone now, and now you have the phenols, the hydroxy groups, okay? Okay, and then we have, what do they do? Lithium hydroxide, which reduces the, cleaves the esters. It's a base-mediated ester hydrolysis. So it just makes carboxylic acids. What they did is they had a mixture at this point, and then they did these steps, and then they're able to separate out the two forms afterwards. Then they did HCl ethyl acetate, which removed the Bach group. So Bach is an acid-sensitive protecting group, and that removed the Bach group, and now they're left with the amines. Okay, and then they were able to separate. I forgot how they did it, but now they actually have a pure version of building block 7A, which is the natural stereochemistry corresponding to the natural tubulysin. And then they have one that's the non-natural. So and they actually, as you'll see, they use this one as well. It's pretty cool. At the very end, they'll use 7A, and they'll also use 7B, and they'll make two different versions of tubulysin. One with a completely natural stereochemistry, and one with a wrong stereocenter at this C terminus of the molecule. And what's crazy is it's still active. It's still just as active as the other one. So that shows that this stereocenter doesn't really matter in terms of tubulysin, in terms of the natural product and its biological activity. Okay? All right, so then let's start with fundamental starting material number 2. Let's go back to see what it looked like. So now we're going to start here and kind of work our way back upwards. Okay, start with this amine, this primary amine. Okay. Okay, so starting from synthesis of from fundamental starting material number 2. What they do is they actually used a reference that there's a sequence, and they didn't show it, and it's not that big of a deal. The main thing is they're attaching a piece with the azide, so it's probably just forming a bond, maybe the leaving group on a carboxylic acid or something. So that kind of attaches here, and you get the azide. And then they're also converting the acetate to a TES. TES is a triethylsilyl group. It's an oxygen with a silicon, silicon, with 3 ethyls. So this is a common protecting group for alcohols, and they decided, I guess, that maybe the acetate it's a little bit sensitive, so it can fall off pretty easily. Esters are really easy to cleave accidentally. And I think in their synthetic strategy, they decided, let's make it into a more stable protecting group. And Tess requires a little bit more work to take off. And this is an ester, and this is an ether. And ethers are generally more stable. Okay. All right. So then they used a strong base, and this ripped off the NH. I think it's like something like LDA, or like a pretty reasonably strong base. It rips off the NH and leaves them with an N minus, and then that attacks the carbon and kicks off the chlorine. Okay. Mechanistically, I think actually the oxygen first kicks off the chlorine, and then the negative charge attacks. And anyway, you get this kind of aminal linkage. It's a weird, kind of unusual linkage, I guess, but it's part of this drug warhead. Okay, so that's how they create this CH2 and then this 4-carbon piece. And then they're ready for their pretty crazy H2 palladium reaction, which reduces the azide, and then they have it in the presence of this another of these the pentafluorophenol leaving group. So this thing is ready and activated for attack. It's an electrophile, so it's ready to be attacked. H2 palladium turns this into a nucleophile, makes NH2, and then the NH2 attacks here, kicks off the leaving group, and then they get this. So that's how they built that molecule. Okay, and then they there's a couple steps that they step through in the paper, but essentially all they're doing is deprotecting Tess and deprotecting methyl ester. So they take off the Tess group, they take off the methyl ester, and then they reacetylate the alcohol. So they need that. The acetyl is kind of a crucial part of the drug for biological activity. So they need the acetate. So it kind of, strategically, they started with acetate, they made it into Tess, they did a bunch of steps, and then later they turned it back to acetate. I think that just made it a little more of a, I think that's what they had to do. That's what they had to do. Okay, so now we have starting material, the kind of sequence number 1 and sequence number 2. So we have the two pieces that are crucial to the synthesis. So let's do that part now. So synthesis of final product, and also its stereoisomer. The word we use if there's a single stereoisomer difference is called epimer. So epimer refers to it's a diastereomer with a single chiral center difference. Okay, so first thing they did is they take it's called molecule number 13, and they do a carbodiimide reaction, and you learned about this in my class, which essentially they're using DIC, diisopropyl carbodiimide. Of course, what does that do? The nitrogen is a base. Nitrogen takes a proton off, makes an O minus. Then the O minus attacks DIC again on the carbon, right? So this takes the proton, then this attacks DIC. That makes a good leaving group on the carboxylic acid. So now you have a good leaving group there, and then the pentafluorophenol kicks that off. Okay, that makes sense? So first you make leaving group number 1, and then this attacks and makes leaving group number 2, and then that makes this pentafluorophenol ester. It's a well-behaved leaving group. And what's really cool about this is once they take this molecule, they can attach the other amino acid component without protecting groups. So this will react with amines, but often when you're doing peptide chemistry, it's like you need a fully protected molecule A and a fully protected molecule B, and then you make the bond and then you deprotect. This allows them to kind of get away with a little bit less, and it's cool. We call it protecting group free peptide coupling. That's what they do here. So this shows the two routes, and these are the molecules we made earlier, 7A and 7B. Remember those? so the amines with the kind of tyrosine-looking thing with the hydroxy, we're abbreviating that. I'm showing a top route and a bottom route here. So in the top route, which makes the top product, the amine kicks off the pentafluorophenol and makes tubulysin B. So there we go. We finished it. We finished the synthesis. And then on the bottom route, it shows this molecule, the stereoisomer with the different CH3 stereochemistry. Nitrogen attacks, kicks it off, and then you get this molecule. And the names of these molecules are called tubulysin B and like epi-tubulysin B. I think they used a slightly more elaborate name, like they defined the position of they give a number for the center that was epimerized. But this is simple. This makes sense. Tubulysin B and epi-tubulysin B, and the only difference is this chiral center. Okay. Okay, and this was interesting in this paper, and it's a pretty readable paper. You should check it out. Interestingly, natural tubulysin B and epi-tubulysin B have similar biological activity. So they did a growth inhibition, tumor growth inhibition assay. It's kind of like IC50. They're inhibiting the growth of a tumor cell. And they found them to be, these are the two molecules, with the difference being right there, carbon and carbon. And they found that they used two different cancer cell types. This is a prostate cancer cell line, and this is a colorectal cancer cell line, and they have natural tubulysin B. So they had natural and they had synthetic, and the natural one gave slightly maybe a little more potent, I guess, than what they isolated. There's a little bit of error in these measurements too, though, so I wouldn't worry too much about this. This is also nanomolar, so it's sub-nanomolar, which is pretty insanely potent, right? It's underneath, it's decimals of a nanomolar, and then their synthetic molecule is 1.1 nanomolar, which is pretty good. And then against the other cell line, that's 1.0. And then they did this 2R tube, which is essentially epi-tubulysin B. They called it compound 14. It's basically the same thing, right? It's also very potent against PC3 and very potent against HT29. And this also looked at Taxol just as a comparison, and it's not as potent. And Taxol is pretty well known to be a super cytotoxic anti-tumor drug. Okay. All right. Okay, so this suggests that C-terminal side, and especially that chiral center, might not be that important in binding, and maybe the more important stuff is on the kind of like left side of the molecules. Okay, so we are ready for development of tubulysin analogs as a new class of anti-drug conjugates. This is Thomas Pillow from Genentech and in his lab of researchers. The paper is called Stabilizing a Tubulysin Antibody Drug Conjugate to Enable Activity Against Multi-Drug Resistant Tumors. I'm going to just do this next time, and it'll still be a good long little lecture, and we'll probably just stop at that point. And yeah, anyways, so this is exciting. I would recommend reading the top paper and reading the bottom paper. I'll put them on the iLearn site so you have access to them. And they're both really cool. Top one you should be able to read after we've talked about it. And the bottom one, there's a little bit of jargon here and there, and it's a pretty brutal paper. But we'll step through this all and explain it all, and it's going to be really fun. It's a real example of an antibody drug conjugate that has very notable activity against multi-drug resistant tumors. All right, see you guys.